Welcome, my dear students, to this session. So in this session, first I'll be talking to you about the different chemistries of nucleic acids. So we start with the molecular biology chapter, in which I'm going to talk about nucleic acid chemistry. I'll be talking about the synthesis of purines and pyrimidines. I'll be talking about DNA. What is the structure of DNA, what is the structure of RNA, and then DNA replication, transcription, translation, regulation of gene expression, etc. All these are a part of the molecular biology chapter. Now let us see one by one what it means. Now nucleic acid chemistries in certain books are given separately, DNA structure is given separately, and then replication, transcription. I am going to take them all together. Nucleic acid chemistry, synthesis of purines and pyrimidines, and the degradation, that is nucleic acid metabolism, and then the structure, and then what is happening to DNA. So I'm going to make it combined. At a time you are going to learn about all the different aspects with regards to DNA and RNA. So let us start with the nucleic acid chemistry. So what are nucleic acids? Nucleic acids are acids which were present in the nucleus. We call them as nucleic acids. That is how the name nucleic acids came. So what are they? They are all macromolecules. Basically it is not a single molecule but macromolecules which are present in all living cells, found in the nucleus. So the genetic information, that is the way you are supposed to be, your habits, your looks, your appearance, your hair, your body structure, everything, the genetic information related to a person is present in the nucleic acid, especially it is encoded in the genes. Now the genetic material of living organism is either DNA or RNA. In a way, DNA is the one which is going to form the RNA, so it is mainly in the DNA. And what is DNA? DNA is deoxyribonucleic acid and RNA is ribonucleic acid. What is this difference between deoxy and oxy or ribonucleic acid? Now, if all these contain deoxyribonucleic acid or the ribonucleic acid contain a sugar known as ribose. Now, if we look at it, if there is no oxygen in the second position, we number it as 2' and all that. So this will be 1, 2, 3, 4. So it 5. So 5 is here, 1, 2, 3, 4, 5. It is a pentose. So in the second position, usually hydroxyl group should be there. So if it OH group is there, we call it a ribonucleic acid, or all the sugars contain OH groups, we call it ribonucleic acid. Oxy is removed, oxygen is removed, then it becomes deoxy. This becomes a deoxyribose. Okay, this is the deoxyribose. This is the sugar. If it is present, we call it DNA. That's the difference between RNA and DNA. One basic structural difference. Chemical, what is the difference? Now we keep saying DNA. So what is this and the genes and all that? So is the whole of DNA made up of genes, or are there something else? Actually, genes are the portions of the DNA which encode for a protein. So genes are lengths of DNA that code for particular proteins. They are lengths of the DNA. Do we know the whole of the DNA? Maybe there are non-coding regions are also present in the DNA where genes are not there. They are the non-coding region. Does that mean they are not necessary? No, they are also necessary. But the gene, when we say the word gene, it refers to the segment or the portion of DNA which encodes for a particular protein. Now, what is the basic structure of DNA and RNA?
Both of them are what we call as polynucleotides. So you have to know that both DNA and RNA are polynucleotides. The word is very important. Polynucleotides. If I say what are proteins? Proteins are polyamino acids. So what does it mean? A protein is made up of it is a polymer of many amino acids. DNA and RNA are similarly polymers of different nucleotides. Now they are made up of smaller molecules called nucleotides. So if I have represented a nucleotide like this in a box, now what is DNA? DNA is made up of 2 polynucleotide strands. That is, it will look like this. This is one strand with many nucleotide going on one after the other, and the second strand is also made up of a nucleotide. Hence the word, they are polynucleotides. Whereas RNA is usually a single stranded polynucleotide strands. But basically both DNA and RNA are polynucleotides. Now let us look at, I kept on saying polynucleotide. What is this polynucleotide? Let us look at the structure of a nucleotide. What is this nucleotide? Now a nucleotide is made up of 3 components. It has to have essentially 3 components. Now the 3 components are: 1, it has to have a pentose sugar. It has to have a pentose sugar. It has to have a base. And it has to have a nucleotide will also have a phosphate group. A pentose sugar with a base with a phosphate, we call it as a nucleotide. Now let us go more into details about it. Now what is this, a pentose sugar? This is usually pentose. So it is a 5-carbon sugar. 5-carbon sugar. The sugar in the in DNA is deoxyribose. As I said, if OH is present, it becomes ribose. If O is not present in the second position, it becomes deoxyribose. So in DNA, it is deoxyribose sugar is present. In RNA, it is ribose sugar that is present. So sugar in RNA is ribose. Now, when we are numbering this, to distinguish between the carbon atoms of the base and the carbon atoms present here, these are always numbered with a prime. 3 prime, 2 prime, 4 prime, 5 prime, etc. To distinguish these carbon atoms from the carbon atoms that are present in the base. By convention, these are all primed. We are going to put an apostrophe on top of them. To distinguish between the nitrogenous base, what are the carbon atoms that are there. Now we go further. The second component, as I said, of a nucleotide is the base. So what are these bases? These are nothing but nitrogenous bases, meaning they are nitrogen containing compounds. Which are the other nitrogen containing compounds? Proteins. So other than proteins, the nitrogen containing compounds include the purines and the pyrimidines, which are the nitrogenous bases. Now when a sugar is made with a base, I have shown a base like this, then which are the bases that are present? Now in DNA, 4 bases are present. They are thymine, adenine, cytosine, guanine. Look at the way they are nomenclature. Look at the nomenclature. Adenine, guanine, cytosine, thymine are the 4 bases present in DNA. And in RNA, there are 4 bases. Instead of thymine, uracil is there. The base uracil is there. And what is now this called as? Without the phosphate group, a pentose sugar with a base, we call it a nucleoside. Nucleoside is, this is important, is sugar plus base. Sugar plus base is a nucleoside. And now we go ahead. What are nucleotides? Let us look at the bond between the base and the sugar is a N-glycosidic bond. So let us go further with the structure of the nucleotide. The third group, I said, it has to contain a phosphate group. So a nucleotide is nucleoside plus phosphate. So it is sugar plus base plus phosphate is a nucleotide. Sugar plus base only is nucleoside. Tide is sugar plus base plus phosphate. So the phosphate group. So when it gets a phosphate group, it becomes a nucleotide. Now why are the phosphate group important? Because they link the sugar on one nucleotide onto the phosphate of the next nucleotide to make the polynucleotide. So basically it is a 3' 5' phosphodiester bond. So this phospho, for the nucleotides to link with one another, we require the phosphate group. Now let us go more about these bases. Which are the nitrogenous bases? Basically we say that they are classified into purines and pyrimidines. The purines include adenine and guanine, and pyrimidines include TCU, thymine, cytosine and uracil. Now if you look at the further, what is the nomenclature? As I said, if the base is adenine and it is a nucleoside, you call it as adenosine. Adenine plus sugar, that could be ribose sugar or deoxyribose sugar. Now adenine with a ribose sugar, we call it as adenosine. Suppose it was a deoxyribose that is present, we call it deoxyadenosine. So that is how the nomenclature goes, the nucleoside nomenclature. Similarly, guanine will form, nucleoside will be guanosine, and what will be its, if it is deoxy, it will be deoxyguanosine. All these are SINE, adenosine, deoxyadenosine. Guanine will be guanosine and deoxyguanosine. Cytosine is already sin, so the name changes. Cytosine is the base, so cytosine is not a nucleoside. So cytosine becomes cytidine, and the deoxy form will become deoxycytidine. Uracil will become uridine, and thymine will become deoxythymidine. Now uracil, usually the deoxy form of uracil is not there. Why? Uracil is present in RNA, and thymine, the oxy form, that is the without the ribose. Thymine with ribose is not there because thymine is present in DNA, so it is always deoxythymidine. So thymine becomes thymidine, uracil becomes uridine, cytosine becomes cytidine, guanine becomes guanosine, adenine becomes adenosine, and the respective deoxy forms. Now that is regarding the nucleoside and its nomenclature. What do you call it? The nucleotide. Now when adenine is present, adenine plus sugar becomes adenosine, plus phosphate, it will become adenosine monophosphate. If it is having only one phosphate, it becomes monophosphate. Suppose it is having more than one phosphate, high energy compound, it is having more than one phosphate. If it is having two, it becomes adenosine diphosphate. If it is having one more, it is all straight chains. If it is having one more phosphate, what do you call it? It becomes adenosine triphosphate. And what is adenosine triphosphate? ATP. So this is the basic structure of nucleotides. So adenine becomes the nucleotide adenosine monophosphate, adenosine diphosphate, adenosine triphosphate. Please remember that in adenosine triphosphate ATP, it is always ribose sugar. But in DNA, it will be deoxyadenosine triphosphate, deoxyguanosine triphosphate, or deoxyadenosine monophosphate, deoxyadenosine diphosphate, etc. So MCQ point of view, please remember that ATP, the sugar, is always ribose, not deoxyribose. If it is deoxyribose, we are going to say that as deoxy-AMP, deoxy-ATP, etc. So let us look at guanine. Guanine becomes guanosine monophosphate, diphosphate or triphosphate. GMP, GDP, GTP are the deoxy forms. Cytosine also CMP, CDP, CTP and the deoxy forms. Uridine monophosphate, UMP, UDP, UTP, and thymidine is always deoxy, DTMP, DTTP and DTTP. These are the nomenclature of the different nucleotides and nucleosides. Now let us go to furthermore, which are the biologically important nucleotides. Some of these, till now I was saying, they are all present in DNA and RNA. Some of these are present even without being in a polynucleotide strand. So we call these as biologically important nucleotides. By themselves, these nucleotides have their own function within the body. So let us look at the functions of nucleotides, or the biologically important nucleotides present in the body other than the DNA and RNA. So let us look at, we know that ATP is the main energy currency of the cell. Whole energy of the cell is energy currency. ATP-ADP cycle we have seen. It is the responsible for ATP is formed during all the catabolic pathways, and it is converted, ADP is converted to ATP during catabolic pathways, and it is utilized for various activities within the cell, including protein synthesis, replication, transcription. It is involved in all the different anabolic reactions. So ATP is the main energy currency of the cell. Now one more important nucleotide is cyclic AMP. Now cyclic AMP, it is AMP. AMP means minute it contains phosphate, it is a nucleotide. What is the difference between cyclic AMP and ATP is this phosphate group, instead of being just in connecting only to the 5' group, it will form a link between 3' and the 5'. So if a phosphate group is at the 3', 5', and it is forming a cyclic, we call it is forming a connection between the 5' and the 3', we call it as cyclic. Now what is, if it is cyclic AMP, it means here the base is adenine. Adenine with phosphate is a nucleotide. 3', 5' adenosine monophosphate is cyclic AMP. Cyclic AMP is adenosine 3', 5' monophosphate. Now where is it? Where have you heard of it? Cyclic AMP is a second messenger for various hormones, including FSH, glucagon, LH, PTH, etc. So many of these hormones we will see where all it acts as a second messenger in other classes. Cyclic AMP is mainly one of the second messengers. That is, the hormone is considered as the first messenger. Its intracellular messenger is considered as cyclic AMP. What about other biologically important nucleotides? GTP and GDP are involved in substrate level phosphorylation. And where have you heard of this? Succinate thiokinase. Yes, succinate thiokinase, that is converting succinic acid or succinate to fumarate. Succinate thiokinase, GTP and GDP are involved. That is one of the levels of substrate level phosphorylation. Where else have you heard of GTP? GTP is the energy source during protein synthesis, not ATP. In many places ATP is there, but GTP also acts as an energy source during protein synthesis. You must have heard of, just as the way there is cyclic AMP, there is also something called as cyclic GMP. What is the difference here? If the base is guanine here, then and this phosphate is present in the 3', 5', then we call it as cyclic GMP. Cyclic GMP. Now where is it? This is also a second messenger. So 3', 5' guanosine monophosphate. Now cyclic GMP is also a second messenger. For what? Nitric oxide, atrial natriuretic factor, nitroglycerin. Many drugs act by increasing the cyclic GMP levels within the cell. It can help in the vasodilation of different wherever required. So isosorbate dinitrate, etc., nitroglycerin. All these vasodilators, they all act by increasing the cyclic AMP levels in the smooth muscles, and thereby the smooth muscles relax and we have vasodilation. So cyclic GMP is a very important second messenger. It is the mechanism of action of various drugs is because of cyclic GMP. We go further. What about UDP? Have you heard of UDP? Yes, UDP sugar, UDP glucose. Where is it? It is involved in glycogen synthesis. UDP, any other mannose, UDP with galactose, UDP mannose. Where are they all involved? In glycoprotein synthesis. So again, we require to energize a sugar. We want to give it energy. These phosphate groups are having high energy compounds. Usually when we say triphosphate, we write it as a squiggle bond. They are high energy compounds. So when so much energy is present and the sugar gets energized, it is able to donate and combine and form bonds with some other substance. And hence UDP sugar derivatives act as sugar donors, especially in glycogen synthesis, glycoprotein synthesis. CTP, CTP is required for the synthesis of phosphoglycerides. You have heard of different phospholipids like phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine. All these are phosphoglycerides. So for this phosphoglyceride synthesis, CTP is the one which is going to act as is required during the synthesis of phosphoglyceride. CDP choline is required for the synthesis of sphingomyelin. Remember, sphingomyelin is also a phospholipid, but the alcohol is sphingosine and not glycerol. These all are also phospholipids, but the alcohol is glycerol. Here it is also a phospholipid with the alcohol being sphingosine. So for its synthesis also CDP choline is required. Many coenzymes are nucleotide derivatives. When you say nicotinamide adenine dinucleotide, we say flavin adenine dinucleotide. All these are nucleotide derivatives, and they are high. That much of the formation of a coenzyme and its action, everything requires nucleotide. Nucleotides are required for action of NAD, FAD, etc. for the formation actually. Hypoxanthine and xanthine are involved in nucleotide synthesis itself. We'll see how they are involved in later. Are there any synthetic analogues of nucleotides? Yes, an analog is prepared by altering either the base or the sugar. They are used to control cancer or infections. I'll talk more about this as we go ahead. Basically, when a nucleotide analog is used, it is used to decrease the nucleic acid synthesis by cancerous cell. We'll see. They are highly toxic drugs and affect even normal cell division. So these are the examples of the different synthetic nucleotide analogues. Look at it, 5-fluorouracil. I'll be repeating this again where they are acting also. 5-fluorouracil, 6-mercaptopurine, 6-azathioprine, arbenosyl cytosine, allopurinol, azidothiamidine, which is used in AIDS. I will talk about all these synthetic analogues and what is their actions. You will be learning more about this even in pharmacology. So with this we come to the end of what is the chemistry of different nucleotides, nucleosides and the different bases. In the next session we'll go further about how these nucleotides are being synthesized within the body. Thank you.